So let's start. We thank you everyone for participating in this webinar, this first webinar. And we are very fortunate to have Assistant Professor Jutarop Phetcharaburanin from Department of System Bioscience and Computational Medicine, Khon University, to be with us today. He's also a part of our Hub of Talent in Cancer Immunotherapy. Right now, he's also a chairperson of Khon University Phenome Center. So let me briefly introduce his background a little bit. He got his bachelor and master degree in biomedical science from Royal Holloway University of London, UK, and got his PhD in clinical medicine research in computational and system medicine from Imperial College, London. Okay, and now, as already mentioned, he's served as an assistant professor of system biology. And he has numerous awards despite his very young age. So it's very impressive. Example of his recent award is the finalist of the Distinct Alumni of the UK from British Council and the World Cholangiocarcinoma Research Fellowships from the Cholangiocarcinoma Foundation of United States, and also distinguished award from Faculty of Medicine, Khon University. And right now, his research interest focusing on system medicine in cancer, particularly in cholangiocarcinoma and liver cancer, and host and microbiota crosstalk. And as we know that phenome is very important, especially metabolite and metabolomics. So today we are very fortunate, despite his busy schedule, he's willing to talk to us on the immunometabolomics, bridging immunology and metabolomics. So thank you very much, Professor Dr. Jutarop. The floor is yours.
All right, thank you very much for your kind introduction, and thank you very much to the TTCI and also especially to Professor Natia for having me here today. And of course, I cannot be busy for the TTCI, definitely. And yep, so for today, I'm going to share the concepts or the principles of immunometabolomics. This is the new approach that we can bridge immunology and metabolomics together. And my name is Jutarop Phetcharaburanin, and it's very nice to meet you all today. And hopefully we could come up with a very fruitful discussion later after the talk is done. And this is the overview of my talk today. So I will start with system biology, metabolomics, and its workflow. So basically I will explain what system biology is and how metabolomics can fit in the system biology suite and how that could be conducted in the laboratory, and also some techniques and applications as example in metabolomics, just to give everyone the overview or the conceptualized ideas of how we could apply metabolomics in clinical studies or other studies. And then I will move on to immunology and basic immune cell metabolism. I'll end up my talk with the immunometabolomics and immunometabolic at last. So let's start with this picture. So this picture shows the complex interactions between human and environment. So as you can see on this figure here, us human being is just in this box, which is about, I would say, 1/6 or 1/7 of the total area, right? So inside our body, we've got the gut-brain axis, organ development, endocrine regulation, immunoregulation, and metabolism all together working synchronously in order to regulate our body and to maintain our body at the regular functions. But human body is not just us, right? It's not just itself. So we need to interact, or somehow we don't have a choice not to interact with the environments. So in the environments, we've got the airborne microbiota, which could come with the aerosol or dust, for example. We've got the oceans or water, where we can be in contact with some other marine or water microorganisms. We have somehow need to be in touch with some soil microorganism or some other pollutants, toxins, and so on and so forth, any other xenobiotics as well. So all of which could affect the human body function in such a way. So that's why, in order to understand the overall picture of the complex interactions between us, human, and environment, needs to be elucidated using the high throughput approach. Having talked about system biology, this is a holistic analytical approach. And before, if we look at this picture, this is a very, very classical example that, system biology lecturer would give, this picture as an example to the learners to understand what actually it is. So usually before that, we would look into different parts, of the elephant on its own, or individually. For example, if we would like to know about the ears, we just focus or target on the ears. If you would like to know about the tails or the ivories or the nose, we just, use the tools or the methodology that can target, on that particular part. In this case, if we apply into molecular biology, for example, if you would like to know about one protein, we just study one protein. If you would like to know about one gene, before that, we just study only one gene, right? But these couldn't be, the information coming out from this analytical approach we call reductionism cannot be used to compose the overview, or the overall image of the elephant. So we can't really visualize or at least imagine how the elephant as a whole would look like, by looking at different parts individually. So this has given rise to the science of system biology. So system biology is about putting together rather than taking apart. We call holism or holistic analysis. We talk about integration rather than reduction, as I mentioned. And this requires that we develop ways of thinking about integration that are as rigorous as our reductionist programs. But different, it means changing our philosophy in the full sense of the term. And this has been stated by Dennis Noble. And as you can see on the far right figure here, that the living organisms have different layers of biological information of which they all interact together in order to produce or generate social functions within the living organism. Now, let's move on to phenome. What actually the phenome is. So I'm sure that everyone is very, very familiar with the term genome, right? And phenome, somebody might have a 50%, 80%, or even 100% understanding of what the phenome is. A phenome actually refers to a person's unique fingerprint of their biology that results from the interaction between the genetic and environmental factors. That means, even though we are human, or we are Homo sapiens, right? But still, we come from different genetic background, family, we live different lifestyles, we live in a different geographical areas, we eat differently, we live our life differently. So this could affect the alteration in the genetic materials or genetic factors, and that would refer to the different varieties of phenotype we call the phenome. And this video would probably show you the overview or the concept of what the phenome is. I'm not sure if the sound is there.
There's no sound.
No sound. Okay, let me share again with the sound share. 1 second. Okay.
A phenome. What is it? Simply put, a phenome is a snapshot of your biology. Everybody has a different phenome that depends on the way genetic factors interact with your lifestyle and environment. The factors that create your phenome are also the things that impact your disease risks. So being able to measure your phenome will enable doctors to develop personalised health treatments in individuals and across populations. A phenome.
So as you can see here, different people have different phenomes, right? Or in other words, we could say that different people would have different set of phenomic data. So if we have a look into the different phenome levels, so as you can see here, the phenome is the interaction between the genome and environment, right? So that means if we look into the population size, or organ size, tissue, or even cell. So we start from here. So this is considered as clinical or person level phenome. And when we look into within the cells, for example, if we look into the metabolites, proteins, epigenomes, and transcripts, we call the molecular phenome. And this information is really important in order to serve as the big data pool, or in other words, molecular big data pool that can lead to the personalized medicine. So only clinical or person level phenome alone, or just one single omic alone data cannot, give 100% or the thorough understanding of the personalized medicine. So that means integrating different layers of biological data is essentially required in order to understand the disease progression, disease etiology, treatments, and surveillance. So a person's phenome is actually a dynamic fingerprint of their unique biology resulting from the complex interactions between environments and genetics. And this has given rise to system biology, as I mentioned. So when we look into just one single omic, in this case, which is a genome, this can tell us what can happen. But when we look into transcripts, it tells us what appears to be happening. But when we have the proteome data, that would indicate what makes things happen. And for metabolome, it tells us what is happening right here, right now. So that means the metabolome is closest to the phenotype that we could observe through our sensation, eyes, ears, or touching, or even some medical equipments that we use for detecting clinical parameters. And the metabolome, in particular, I would like to point out this term because this is one of the key terms in this talk. So metabolome comes from two terms, metabolo, which is from metabolite, and ome means entire or all. So this is the size of the study of entire metabolites or small molecules that have the molecular size of the molecules of less than 1,500 Daltons. And when we apply the metabolomics into clinical studies, we call clinical metabolomics. What are those molecules that we would look into? So for clinical metabolomics, we would look into amino acids, cholesterols, and derivatives. We would look into neurotransmitters, bile acids, some sugars, free fatty acids, vitamin and cofactors as well, and so on and so forth. So these molecules can be detected using metabolomics. And this schematic diagram shows the applications of metabolomics. So people are very familiar with metabolomics use on the biomarker discovery, but not only biomarker discovery that can be utilizing metabolomics. Metabolomics can be employed for pharmaceutical R&D, nutrition, consumer goods QA and QC, bioprocessing and cell culture science, animal health and nutrition as well, aging and gerontology, and some other medical applications in both cancer, microbiota science, neuroscience and neurology, phenotyping and genomics, and metabolic and cardiovascular diseases. When we talk about metabolic phenotyping, there are quite a few factors contributing to the changes of the phenotype. And these include genetic and epigenetic factors, age, environment, diseases, drugs, nutrition, and lifestyle. So all of which can be reflected through the phenotype that we could observe. So that means the phenotype can be monitored thoroughly using metabolomics. And the metabolome data can lead us to the precision medicine in two arms. One is for health monitoring. In that case, we can look into the nutrition, lifestyle, gene-diet interactions, for example. And the other arm includes the disease risk prediction. And also we can use metabolomics to estimate the efficiency of drug treatment as well. The techniques used in omics. So if it's for genome transcriptome, of course, we need to use sequencers, and there are quite, very, many kinds of sequencers nowadays, short read, long read. It really depends on the purposes of the research study or the research questions. And for proteome, we use mass spec, of course, and metabolome, when we need to analyze these small molecules, for example, amino acids, lipids, carbohydrates, and some xenobiotics, for example, drugs. We use actually there are three major platforms for metabolomics: nuclear magnetic resonance spectroscopy or NMR, mass spectrometry, either liquid chromatography mass or gas chromatography coupled with mass spectrometer. And in metabolomics, there are actually two approaches in order to conduct such a study. One is called targeted metabolomics. For targeted metabolomics, we need to have the metabolites of interest, or those molecules that we have already known, that they are the drivers of the disease, or they are the disease biomarkers, they are treatment or monitoring biomarkers, for example. And it is a hypothesis testing strategy, and it requires a validation of potential biomarkers. For untargeted metabolomics, this is when we perform the global profiling. That means we have no ideas what metabolite would be the key players in our study. So we have actually no clue. So we start from scratch. And it is hypothesis generating. So that means when we perform the untargeted metabolomics, the data obtained from this study would open up new hypotheses of which we would know or we would not know at the beginning. So sometimes it would help us open the new hypothesis leading to the newer research questions in the field. And it is correlated with database, and it is used for biomarker discovery. And this flow chart shows the workflow of the metabolomics. So in metabolomics, just like other omics study, we start with the experimental design. And this is the most important and most crucial step in any omics studies, because actually, I used to have some questions from collaborators or researchers who have been working together at first after I came back from the PhD. So some of them asked me, would it be okay to run the serum metabolomic studies using my archive samples? And I asked them back, how long have you kept them for? And then they said about 5 years. So 5 years is quite long, right? It's quite old samples. And also, when I asked for further information about the sampling method, about the containers that were used for sample collection and so on and so forth, it seems like the conditions conducted for this sera samples is not appropriate for metabolomics. So I suggested them not to waste the money and run their samples on the metabolome analytical platform, because if we get the rubbish in the machine, we will get the rubbish out as the rubbish data. So that would not be interpreted anyway. So experimental design for omics study is really important in order to reduce the potential confounding factors, and also to avoid any bias that would be introduced into all the steps in omics studies. And then we started sample collection after we obtained a very clear and nice experimental design. And after that, we need to prepare the samples according to the platform that we are going to use. If it's NMR, it has a specific requirement for sample preparation. If it's the MS, it has the other way of specific requirement for preparing the samples. And then after we obtain or acquire the data, we need to pre-process the data in order to make it comparable across all the samples in the study. And again, the techniques for pre-processing actually depends on the machine or the instrument that we acquire the data from. So if it's NMR, there is 1 or 2 pipelines, and if it's MS, there are quite a few pipelines that we can follow, for example. And then after we pre-process the data, we need to perform the data analysis and perhaps do the integration and network analysis as well, in order to understand the overall metabolic alteration as the big picture, in our study. And then we can identify the key metabolites that make let's say the treatment and the control groups differ from one another. Or we could use the metabolite identification in order to specify the biomarkers that we found from the statistical analysis. And in the end, we need to interpret our data and validate our data. For validation in metabolomics, there are 2 ways. One is to do statistical validation, where we use a statistical approach in order to validate the validity of the model. Or we can perform the biological validation, where we run another or parallel study and collect another set of samples and run them exactly the same way as we have run the first batch of results and use that data to validate our first batch of data, for example. And in order to analyze the metabolome data, we use an approach called chemometrics. So chemometrics is the combination of computer science, applied mathematics, and multivariate statistics all together in order to understand and elucidate the meaningfulness of the data, not only from metabolome analytical platforms, but from the DNA RNA sequences as well. So, the high throughput instrument that we use for characterizing each omics, these platforms actually generate high throughput data, which require the chemometrics, as a tool to analyze these data. And this would be beneficial for many fields of study: agriculture, environment, chemistry, medicine, biology, and also in industry as well. And for chemometrics, so this is a use of the mathematical and statistical methods, and this can be used to decide or select optimal measurement and procedures and experiments at the beginning before we obtain the study design. And it's also to provide maximum chemical information by analyzing chemical data. So this is after we acquire the data already. So that means the chemometrics is very important in order to make the very best use of our omics or metabolomic data in this case. And yep. So this is the flow of chemometrics. We start with data in order to generate information, and this information can help generate the knowledge accordingly. And this slide shows the characteristics of the metabolome data. So metabolome and other omic data is usually high throughput. That means the high throughput data would usually have the high dimensionality, for example, many variables per one samples, right? A degree of collinearity. And also, we can't really expect to be able to identify all the signal features coming out from the analytical platforms, either NMR or LC-TCMS, because most of which are unidentifiable. So that means just only maybe 50 or up to 70% that can be identified on NMR, or less than 50% on MS that could be identified. And it requires the multiple analytical methods in order to confirm the existence of such metabolites or small molecules. And last but not least, the metabolomic data has a very high sensitivity to many effects, for example, errors, bias, confounders. So we need to be highly aware of these biases, that may arise by the introduction of some inappropriate sampling method, for example, or sample prep method, or unsuitable pipelines for data analysis. And the examples of the metabolomics that was used in such cancer study is in cholangiocarcinoma. So as you know, cholangiocarcinoma or bile duct cancer is the epithelial malignancy of either intra or extrahepatic biliary tract. And this is the second most common liver cancer, following the hepatocellular carcinoma, and it accounts for 10 to 15% of all hepatic cancers. And this slide shows the global incidence of the cholangiocarcinoma. And as you can see here, in Great Mekong sub-region countries, especially southern China, Thailand, Cambodia, Myanmar, and Laos, have a very high incidence as represented in red color. In the northeast of Thailand, it has about 85 per 1,000 people. So the highest incidence of the CCA has been reported in the Southeast Asia, and this is still the major health problem in Thailand, particularly in the northeastern region. And if we have a further look into the northeastern region, here, Khon has the OV infection of about 118.5 per 1,000 people. So in GMS countries, we found that the OV or Phetcharaburanin, the liver fluke, is a major cause of cholangiocarcinoma in this area. And this is the video showing the cross-sectional liver that the surgeon in our research institute has removed. And as you can see here, this is the liver fluke that are swimming around or within the bile duct, dilated bile duct. And this shows the liver flukes obtained from the patients. So far, in regards with the cholangiocarcinoma screening and care program or CASCAP, led by our former director, Professor Narong Kantigao, and our current director, Associate Professor Vacharin Loilom, and together with other multidisciplinary research staff from different departments in faculty medicine and other faculties of KKU and our collaborative universities. So we have developed the gold standard for the early stage detection of cholangiocarcinoma, and this method has been developed and optimized by Professor Nitaya Shamadan, who is the radiologist in our Senior Korean hospital. And as you can see on the top panel here, this is a normal echo pattern that shows the normal bile duct, whereas the bottom panels here shows the abnormal echo pattern, where we can see the starry sky, and the dilated bile duct here. So far, we use this technique in order to screen the population in the endemic areas. And also, it is quite a very robust technique, but still, in order to move or to remote the ultrasound machines to different area is not an easy way to go. And also, it requires such a specific type of air conditioned room, and also the specialists in order to read the image, right? To analyze the image. So that means biomarker is still needed in this field in order to differentiate people with cholangiocarcinoma or people with high risk of cholangiocarcinoma away from the people with the normal bile duct or healthy population. So this slide, I just would like to give you the overview of the latter research example that I would give in this talk. So basically, in our human body, we have the host nutrient microbe interactions, right? So in our GI tract, it harbors millions and millions of microbes, right? And these microbes play very vital roles in several metabolic functions of us as a host. These include dietary choline metabolism, non-digested fiber fermentation, protein digestion, fat digestion, and bile acid metabolism. So all of which, generate the bioactive metabolites or the functional metabolites that can play, some key roles in many organs and regulate the functions of the organs as well. So biosynthesis is the target of this study that I would like to give as an example. So primarily bile acids are synthesized from the cholesterol in the liver, and some primary bile acids are converted to secondary bile acids by the enzyme activities in the gut bacteria. In the liver, primary bile acids are synthesized through two major bile acid synthetic pathways: the classic pathway, also called the neutral pathway, and the alternative pathway, also called the acidic pathway, due to the production of acidic intermediates. And the bile acid biosynthesis, as I mentioned, there are two major pathways, right? And in the classic pathway, the cholesterol is converted to 7-alpha hydroxycholesterol by the rate-limiting enzyme CYP7A1, which is located in the endoplasmic reticulum of the liver cells. And the 3-beta hydroxysteroid, the hydrogenase, converts the 7-hydroxycholesterol to the 7-alpha hydroxy 4-cholesterol 3-on, which is converted to the 7-alpha and 12-alpha dihydroxy 4-cholesterol 3-on by a sterol. So this is just one example of how the bioconversion of the primary bile acid takes place in the liver. So in this particular example of study that I would like to share with you guys today is the fecal metabolic alteration in normal versus PDF and CCA. So in this particular study, we recruited the participants who have been diagnosed with PDF using ultrasonography and also to CCA with the MRI and CT scan, and in comparison to the individuals with normal bile duct group. And the method that we use for collecting the fecal samples actually derived from our previous publication in 2016, where we optimized the fecal handling strategy, and published in the Analytical Chemistry, ACS. And yeah, we just adopted this pipeline in order to obtain the samples. And yeah, this slide shows the demographics of participants. So in the actual study, we need to exclude some participants out of our study because they fall into the exclusion criteria, and we ended up with 20 normal, 20 PDF, and 15 CCA in this case. So first of all, we performed the global proton NMR fecal metabolic profiling on the 3 different groups of the fecal samples obtained from the participants. And we found that in the fecal samples, it has about 40 fecal metabolites that could be subdivided into 10 classes, 10 chemical classes as shown on figure A and B. And then we perform the principal component analysis, or PCA, on the fecal metabolome data to reduce the dimensionality of the metabolome data and to see the trends and overview of the metabolic alteration across different groups. And then we use exactly the same data to perform the heat map and hierarchical clustering analysis in order to explore the fingerprints or the unique fingerprints of the metabolome data in different groups. So red represents CCA, green represents normal, and PDF has been represented in blue. So as you can see here, the red, the hot color tones in the heat map shows the higher concentration of the metabolites, where the cooler tone shows the low concentration of the metabolite. And for each group of the participant that we study show the different fingerprints of the fecal metabolome concentrations. Then we perform further statistical analysis in order to come up with the good biomarkers that can indicate different groups of study. And then we came up with these 8 metabolites that shows the promising sort of power in order to differentiate the 3 groups of study. And in the end, we managed to come up with a schematic diagram that can indicate the gut health and the gut metabolism that occurs throughout the progression of the disease from normal to PDF and to CCA. So, for example, we can see that the host microbial co-metabolites occur in a higher or increasing concentrations, ranging from normal to PDF to CCA. And some of these metabolites have been particularly generated by the proteobacteria or the gram-negative bacteria. And from this study, we showed that the global metabolomic in feces exhibited gut health status and host microbiota interactions during carcinogenesis. And some other inflammatory metabolites are also increased in the PDF and CCA as well. So at the end of this paper, we did make some suggestions to the clinicians who look after the CCA patients to further monitor their gut health as well, as per our findings that we found there is a likelihood to have the development of the gut inflammation thereafter. Another study that we performed is to look into the bile acids in the feces. So as you know, that the after the CCA patients have the obstructed biliary tree, so the bile cannot go through the lower GI tract, So that means the ecological niche in the lower GI tract would change, especially the pH, and this would affect the population and function of the microbiota. And that would in turn lead to the different or altered bioconversion of bile acids from primary to secondary bile acids as well. And in this particular study, we managed to optimize the UHPLC-MS method in order to differentiate and detect 32 fecal bile acids for both qualitative and quantitative analysis in a 7-minute method. So that means per one sample, we managed to have a profile of 32 bile acids in 7 minutes, and that could be used for further diagnosis straight away. So in these 3 groups of study that we have recruited the participants in, so we have the normal, PDF, and CCA, as you know. So the majority of the bile acid is CDCA, followed by DCA and LCA. But the 4th most abundant of bile acid in normal, in comparison to PDF and CCA, started to differ from this step. So in normal, it's NuPCA. In PDF, it's NuPCA, whereas in CCA, it's a HDCA. And then when we further perform the analysis, we classify our bile acids into 4 different groups. And these include primary unconjugated bile acids, secondary unconjugated bile acids, primary conjugated bile acids, and secondary conjugated bile acids. And we found that from normal to PDF to CCA, it shows the decreasing trend of the primary unconjugated bile acids, and of course, the increasing trends of secondary unconjugated bile acids. And then we perform the heat map and hierarchical cluster analysis to visualize the unique fingerprints of bile acids in the fecal samples obtained from these 3 groups. We found the distinct bile acid signatures of CCA, normal, and PDF. And then we perform the supervised analytical method in order to extract more biomarker discovery information. And as you can see on figure A here, the scores plot of the OPLSDA shows the very clear separation of the 3 groups from normal to PDF and CCA. And then we perform further statistical analysis in order to see the power of, discrimination of these bile acids. And we ended up with the 10 bile acids that could be used for checking with the sensitivity and specificity. And we managed to obtain the panel bile acids that can show 90% of specificity and 100% of sensitivity in this case. So certain bile acids show the potentially high sensitivity and specificity as biomarker panel for discrimination of PDF and CCA from normal groups. So this, all of these findings have been put into a manuscript, and it is in preparation for further submission. So that's it for metabolomics. So hopefully by now everyone would have the same pace on metabolomics. And then if we apply metabolomics in order to study the function and behavior of the immune cells, then we can have this term into the track, immunometabolomics. So for immunometabolomics seeks to understand how the metabolism of immune cells change during different activation states or in the different environments. It also aims to understand how these metabolic changes can influence the function and behavior of immune cells, and also the roles of the specific metabolites in modulating immune responses. And last but not least, immunometabolomics help us understand the potential to therapeutically target metabolic pathways to enhance or suppress specific immune responses. So this figure shows the immune system that consists of a complex network of cells, tissues, and molecules that work together to defend the body against harmful pathogens and diseases. Different immune cells like T cells, B cells, and the macrophages play specific roles, and their activity that can be influenced by various factors, including their metabolic states. And several principles have recently emerged in immunometabolism. Extensive interplay exists between metabolism-associated environmental cues, for example, nutrients, and immune receptors. which act in synergy to mediate innate and adaptive immune responses. The outcome of immune reaction is further shaped by the crosstalk between signal transduction and metabolic reprogramming in a process known as bidirectional metabolic signaling. And such metabolic signaling serves as a driver of immune cell state and fate and tissue immunity, evidenced by some recent findings in T cells, B cells, and dendritic cells, and also macrophages, among other cells. And finally, as metabolism function as highly complex networks for both cell signaling and cell-cell and cell environment interactions, there is actually the increasing appreciation of intracellular and intracellular networks for immunometabolism. Therefore, understanding immunometabolism at the signaling, cellular, and systems level has advanced our understanding of immunity and metabolism, and also provided new opportunities for therapeutic targeting and immunomediated diseases, cancer, and other metabolic disorders as well. So this schematic diagram demonstrates the metabolic changes in immune cells based on inflammation status. Glucose, as you know, is a primary or principal substrate used by the effector cells such as neutrophils and M1 macrophages for the generation of energy, right? And these are also the cells which are the first responders to initiate an inflammatory response and thus pro-inflammatory in nature. Fatty acids, on the other hand, is involved in the fatty acid oxidation, which is the predominant energy generation pathway used by the naive and tolerant cells such as M2 macrophages and T memory regulatory cells. And these cells are generally involved in resolving the inflammation and thus are anti-inflammatory in nature. So this figure shows the metabolic differences between normal cells in figure A, and the cancer cells in figure B. And the normal or quiescent cells in aerobiosis metabolize glucose mainly to pyruvate, which is then oxidized in the mitochondria to carbon dioxide using the TCA cycle and OXPHOS for generation of ATP. And the ATP synthesis takes place preferentially in the mitochondria. In anaerobiosis, pyruvate is metabolized to lactate instead, and cancer cells in anaerobiosis or anaerobiosis converts most glucose to lactate, which is exported to the TME or tumor microenvironment, while diverting some glycolytic intermediates to the PPP, which generates NADPH and pentoses for the synthesis of nucleic acids. ATP synthesis is actually largely cytosolic, as you can see. So glutaminolysis generates glutamate, which is converted to alpha-ketoglutarate, a major substrate to refuel the TCA cycle. And that means the TCA cycle intermediate citrate, in this case, is exported to the cytosol, where it is converted to acetyl-CoA, used for synthesis of lipids. So in cancer cell metabolism and derangements in the tumor microenvironment, the mitochondrial oxidation of nutrients, including glucose, amino acids, and fatty acids through the tricarboxylic acid cycle and the electron transport chain, is a highly efficient means of producing energy for quiescent or differentiated cells. However, during periods of increased proliferation, such as after immune activation or malignant transformation, the cells upregulate an alternative pathway for glucose metabolism called aerobic glycolysis. Although less efficient in generating ATP, aerobic glycolysis allows for more rapid metabolism of glucose, right? And efficient disposal of excess carbon and regeneration of the NAD+, while preserving mitochondrial enzymatic activity for anaerobic processes. And the glycolytic intermediate are channeled through the other essential pathways, such as the pentose phosphate pathway or PPP, as I mentioned, and the 1-carbon pathway and the hexosamine biosynthesis pathway, and so on and so forth. So this slide would probably give you some example of the adoptive T cell therapies that enable flexible approaches to metabolic therapy. So the T cells in such regimens can be genetically modified to express the chimeric antigen receptors, or CARs, as we know, that recognize known tumor surface antigens and trigger the T cell receptor signaling in the absence of measured histocompatibility complex presentation. T cells can be metabolically conditioned through the use of the chemical inhibitors or genetic editing during the ex vivo expansion through the metabolically engineered media or through the pharmacologic treatment of the T cell reinfusion. Many of the interventions discussed here in this picture, including inhibiting glycolysis, glutamine metabolism with 6-diazo-5-oxo-inorleucine, or DON in this case, and AKT mTOR signaling and potassium supplementation, have been used in the ex vivo T cell expansion and led to the improved T cell persistence and anti-tumor response. And some recent studies actually demonstrated that the forced expression of the peroxisome proliferator activated receptor gamma co-activator 1 alpha, or PTC1 alpha, which promotes the mitochondrial biogenesis in adoptively transferred CD8 plus T cells, resulted in the superior intratumoral metabolic and effector function as well. So using small molecules, monoclonal antibodies, and genetic editing metabolic processes can be targeted to either disable cancer or suppressive immune cell metabolism, or conversely engage and support effector cell metabolism. The metabolic processes in the suppressive immune populations and cancer cells can be targeted to directly decrease viability as well as to disable the metabolic pathways that deplete nutrients. For example, arginase 1, as you can see as ARG1 here, and the indoamine 2,3 dioxygenase, or IDO. These can lead to the toxic metabolites, for example, lactate and CD73, or induce metabolic control of effector cell populations, for example, the mutant IDH1 generation of the oncometabolite 2-hydroxyglutarate, or R2HG on this picture. Metabolic interventions may also be able to induce beneficial changes in the effector populations, such as increasing longevity and antigen-specific immunologic memory. So this slide shows the immunosuppressive effect of lactate and extracellular acidification in the tumor microenvironment. And the tumor, as well as immune infiltrates and stromal components of the tumor masses, contribute to different levels to the overall lactate and proton pool in the cell, in the tumor, and also in the tumor microenvironment. Lactate and acidification provide the metabolic support to sustain tumor cell growth. And at the same time, they actually dampen anti-tumor immunity by inhibiting effector immune cell populations while boosting the suppressive immune subsets. Stromal cells can contribute lactate that can be taken up and oxidized by the tumor cells, or we call reverse Warburg effect, or the regulatory T cells contributing to the anabolic metabolism. And this schematic diagram shows that lactate metabolism is tightly intertwined with epigenetic programming in immune cells. As you can see here, the aerobic glycolysis and lactate production regulate the immune cells differentiation by influencing the activity of the epigenetic machinery. So this is this example of targeting aerobic glycolysis in tumor and immune cells to boost cancer immunotherapies. So figure A here shows the inhibition of the glycolysis-inducing pathways, where Warburg metabolism is driven by mTOR and or the interconnected HIF1 alpha signaling pathway downstream of oncogenic signals. The blockade of these signals cascade can be used to inhibit the aerobic glycolysis in the tumor cells to curb tumor growth. And Warburg metabolism is also induced in immune cells in response to pro-inflammatory and costimulatory signals, in this case, the toll-like receptors and CD28, that trigger the PI3K AKT mTOR axis and thus targeting these pathways unselectively can result in diminished immune anti-tumor effector responses. And on figure B, this shows the targeting lactate production and acidification of the tumor microenvironment to counter immunosuppression. As you can see here, the LDHA inhibitors, or indicated as the LDHAI in this picture, are being developed to block the production of lactic acid from pyruvate. That means lactate export can be blocked by specific MCT1, MCT4, or dual MCT1/4 inhibitors as well. And proton and potassium ATPase inhibitors reduce the release of protons to the extracellular space. Bicarbonate infusions can be also used to neutralize protons in this case. And carboxylic anhydrase extracellular produce the protons and bicarbonate, which is used for the intracellular pH buffering as well. So these strategies, in addition to the repressing anti-tumor immune responses, can have direct anti-proliferative effects on both tumor and T cells. And figure C here shows the reversing lactate-induced rerouting of pyruvate into the TCA. So lactate reduces pyruvate anapyrosis via the pyruvate carboxylase, or PC, and autocrine succinate signaling. So inhibiting pyruvate dehydrogenase restores the PC activity, succinate secretion, and signaling, as well as CD8+ T cell cytotoxicity as well. And last but not least, figure D here shows the targeting aerobic glycolysis to enhance T cell fitness for adoptive cell therapies. So this schematic diagram shows the direct and indirect inhibition of the aerobic glycolysis by small molecules, cytokines, transmembrane ligands, overexpression of the metabolic regulators, or by the selection of the chimeric antigen receptor constructs that the engaged oxidative phosphorylation can be used to promote the generation of stem-like T cells that exhibit increased anti-tumor function. So as you can see here, this is where and when the immunometabolics can jump in order to prove the functions and behavior of these immune cells that are involved in such different steps in immunotherapy. So the last part of my talk today that I would like to introduce to everyone is the Immunometabolic Atlas, which is the tool that some group of scientists that created. So currently there is a lack of the tool to determine known associations between metabolites and the immune processes. So therefore, interpretation of metabolites in the metabolomic studies in terms of the role in the immune system, or selection of the most relevant metabolite classes to include in the metabolomic study, is challenging. So this study, published in PLOS One, actually described the Immunometabolic Atlas, or IMA, which is a public web application and library of R functions to infer immune processes associated with the specific metabolites and vice versa. So that means if we obtain the metabolome data of the immune cells, right? So we can input our data into the IMA, and that would generate the associated metabolic map in order for us to see or to visualize the overall alteration of the immune cells functionality, and also the behaviors, and also the interactions within different immune cells as well. The IMA here provides the association between the metabolites and immune processes of interest through the generation and evaluation of the protein metabolite interaction network. So it's pretty easy to use as long as we have the more than enough metabolome or protein data. So this slide shows the overview of the information and associations retrieved from available databases that the IMA use. So they use quite a few databases, including Gene Ontology, UniProt for immune processes and proteins respectively. They use Human Metabolome Database for the metabolites pool. They use String, and rare as well for protein-protein interactions and metabolite-metabolite interactions in this case. And on figure B here, it's a study flow to build the protein metabolite interaction networks to associate metabolites and immune processes of interest. So that means, as I mentioned, as long as we have one, or the other omics data between metabolome or proteome, we can input, this and the interaction between the protein metabolite and immune cells together in the metabolic map. So in summary, hopefully after this 45-50 minutes, I wouldn't frighten you guys with this information or bombarding you guys with this information, but hopefully we would have the same or similar picture of what metabolomics is and immunometabolomics, how we can how we can employ immunometabolomics in this immunotherapy research. So metabolic profiling at first is the holistic and analytical approach at the molecular level that can help enhance the systematic visualization of health status, biomarkers, and treatment monitoring. And this approach is to deal with molecular big data combined with clinical data. That means in order to maximize the meaningfulness of data, we need high performance computing facility, for example, and the chemometrician who are the scientists who works on the chemical data in order to lead us to personalized medicine, or the true personalized medicine, I would say. And last but not least, immunometabolomics provides a new lens to understand the immune system by studying its metabolic underpinnings, which can be crucial for both disease understanding and therapeutic development. And finally, I would like to thank my young investigator grant mentors, Professor Jutarop from Mahidol University, Associate Professor Wacharin Loilom from Khon University, and Associate Professor Jelly from Imperial College London. And my colleagues back at in Khon University, at Imperial College London, Australian National Phenome Centre, Murdoch University, and our multidisciplinary research teams at KKU who help conduct many studies that have been completed. And I would like to thank the funders, National Research Council of Thailand, Cholangiocarcinoma Foundation of the United States, Royal Society of Chemistry of the United Kingdom, and Cholangiocarcinoma Research Institute, and also the Ministry of Higher Education, Research, Innovation, I forgot one thing, Science, sorry, it's a very long name. And just a quick announcement. So at the KKU Phenome Center, we work together with Sivila along with Lab. We are going to organize the multi-omic short course during the 18th of January to the 2nd of February next year, 2024. And the application is still open for those who are interested. So please feel free to scan this QR code to our KKU Phenome Center Facebook fan page for more details, or you can just drop us a message, or just give us a call if you have any other questions. So yeah, feel free to contact us, or you can add the live OA as well, just for a quick response. Yeah, so in this short course, we will introduce to the participants about the study designs and all the hands-on experience on genomics, transcriptomics, proteomics, metabolomics, CRISPR-Cas, and metagenome analysis. So all in one package. So this is a very good chance, to come and join us. And for those who have been selected, you will be provided with a full scholarship supported by PMUB. So this is not first come, first serve basis, but it's on the selection criteria. But please feel free to apply. And hopefully we can meet each other in January next year and work together thereafter. Yeah, thank you very much for your kind invitation and having me here today with the TTCI and all colleagues and collaborators. Thank you very much.
Wow, thank you very much, Dr. Jutarop, for such a comprehensive review to us. We are really appreciate it. So Dr. Jutarop has already telling us how important to look at the metabolite in the microbiome as well. Like we're very familiar with 16S ribosomal sequencing, but actually to look at the metabolite in the stool or even in plasma, it's also important. So he showed us his results using NMR and mass spec. And then at the end, he's telling us, which we already know, that's why we invited him, that metabolites also, influence the T cells, the immune cells. So that's why we're very interested in this. So I would like to invite any questions from the audience to ask Dr. Jutarop. Are there any let me ask, and I know you haven't done any immunometabolism in the cell, in the immune cell before, right? Is the technologies the same? Do we use NMR and mass spec? Can you share that detail with
Sure.
us?
That is a really good point. So basically the major platform for analysis of the metabolome data remain the same. Well, we can use either NMR, LC, or DCMS as the key platforms, but the sample preparation would have been different because in order to sort the different cell types into different sample preparation procedure, that is a key for understanding different cells functions and behaviors. So that means cell sorting must come into action for applying metabolomics into immunometabolomics in this case. Yeah.
So we do have to sort the cell, the cell of interest to do the analysis, right?
In this, in the context of immunometabolomics, yes.
How much cell do we need?
Well, actually, this is a very good question as well. So if it's for NMR with the resolution of 600 megahertz upwards, I would say about 500,000 would be enough. Yeah, but if we use the mass spec or either GC or LCMS, we can use pretty much lower, maybe 100,000 cells should be enough. Yeah, but it really depends. So the next question that people would have in mind is that how do we know whether we should go for NMR or LCMS, right? So this is very classic sort of question that I get asked every time when I give a talk. So I would say that if we don't have any data in hand, we never done metabolomics for such samples or cells, I would suggest use NMR to conduct global metabolic profiling first because of the robustness of the NMR and high reproducibility, and also for metabolite identification of the NMR is over the MS, because usually the signals obtained from NMR we can manage to identify, I think, up to 80 or 70% of metabolites by the NMR features. Let's say if we can detect 100 signals from NMR, about 80 metabolites can be fully identified. But MS, maybe 10 to 20%, and the remaining will be just, the trash. So yeah, if it's for untargeted or global metabolic profiling, I would suggest NMR first. But after we know some metabolites that we would like to target further, let's say if we know some players that contribute to such the phenomenon that we would like to study, we can then use LCMS or GCMS later. Yeah.
Thank you very much, Ka. We have a hand raising from Sahat Chai, right? Yeah, please.
Yes. Thank you very much for such a great talk. Can everyone hear me?
Yes.
Yep.
Yes.
Yes. Okay. I have a very brief and might be a naive question here. I have done some type of work with metabolomics before, and my problem that I encounter is that it's indicating the metabolic pools, but it's not actually. It's quite difficult to refer the metabolic fluxes out of those data, right? Mm-hmm. And when we want to know mechanism, especially in cancer, most of the time, for example, Warburg effect or even the reverse one, we want to see the flux. We want to see the direction of the flux. We want to see the quantity of the flux as it's more like to answer the mechanism more than the metabolic pool itself. So do you have any facility or do you have any plan on doing work further in fluxomic or any other type of data that not just only metabolic pools?
Yeah, that is a really good point. So it is one of the goals that our KKU Phenome Center would move on to, is just to establish the facility for everyone, actually, not just KKU researchers, but anyone who is interested in using the facilities in the future in order to do the flux analysis of the metabolic functionality. So yeah, we are in the process of moving, of getting more facilities in order to prompt us, to be able to do so. So it requires a bit, I think I would say 2 or 3 add-ons instruments that we need to get established. Yeah. So it's a really good point indeed. I understand that, metabolic pools would tell us just some signatures or fingerprints, but in order to visualize the functionalities or mechanism, that would be great to answer to research questions. Yeah, I totally agree.
Yep. So basically the work I'm
Yeah.
working on now will require at some point to do the metabolic fluxes. So I hope in the future I can reach out to you and we probably can collaborate.
Sure. Yeah, we can also do, well, we're now in the process of developing the labeling metabolomics as well. So we are going to label the cells, in order to monitor, the degradation or the bioconversion of that particular molecule into different biometabolism throughout, the study period as well. So that would probably help as well. Thank you for raising this point. Yeah.
Thank you very much.
You're welcome.
Thank you very much for the question ค่ะ I can see another hand raising from Dr. Supanika ค่ะ
Hi. So thank you very
Sawadee ka.
much. Sawadee ka. It was very great and very comprehensive talk, especially for someone that know nothing about omics like me. So I just have a very kindergarten questions about omics things so first question is that can we do metabolomics together with genomics and proteomics, and would that how you relate these data together, and would that be more meaningful when you combine all of them? That's the first question. And then the second question is actually continue with Ajanatia question, in terms of the technique wise, would it be possible that if we could look at the metabolomic of immune cells like T cells or something from serum from the patient, would it be possible that, let's say if we have patients coming in and we want to check a marker if this patient is having a T cell fitness, that we could use to make a good CAR T cells, things like that, without sorting a particular cells and look at cellular level.
Right. Okay. Yeah. So let me address the first question first. So for the integration of the omics data, so this is the most important issue for now in all the research fields that use or employ omics techniques or technologies. So obviously we're gonna get more information, when we are able to integrate the different omics layers, of information, because metabolomics alone cannot tell us the genetic traits, of the cause of the disease, of course. But that means it would be more beneficial for anyone, who works in the field, if we could obtain the genomic, transcriptomic, yes or no, or proteomic and metabolomics all together in order to elucidate the structure of the data, and also to see the flow of the biological data from within the nucleus, outside the nucleus to outside the cells. So that would help answer the thorough questions, many questions would be answered by that. But to do so, it's quite challenging nowadays with bioinformatics community. We are trying to develop the computational tools in order to integrate the different omics data, because as you may know, the different omics data comes with the different data pre-processing protocols or pipelines, and also normalization of data is another challenge that we need to overcome, because different omics data require different use or different means of normalization. So how could we make different omics data more comparable before integrating them together? So that's the only challenge. Well, I would not say the only challenge, but that's the biggest challenge that we need to overcome for now. But I believe that maybe in the next 1 or 2 years, perhaps, because this is a fast growing field, we could come up with the more appropriate way to integrate all omics data together in a fair way, because there's still some controversy between researchers in this field about integrating omics data, and what about normalization, what about data treatment, and so on and so forth. So there are still some minor concerns that we need to address. And for second question, for metabolomic of the T cells, this is a very good, a big step forward. So if we could manage to optimize the method or a protocol that we could, isolate just the T cells, from the cell pool of the specimen, and perform the metabolomics, on analysis on that I think that would be a good way to go, definitely. But yeah, I think R&D is still needed in this stage. So yeah, so if we would like to, have a collaborative project on optimization of this method, I think it would be a great idea too. Yeah.
Wow, thank you very much.
If you look in the serum, you cannot tell where are that metabolite come from, right? So most of the study
it's a
is
mixture
on the sorted cell.
Yeah.
But then, Jutarop, do you think maybe we have to do literature search whether we can
Exactly.
look at the serum or not, right?
Exactly. I would believe that, secretomes, in the aspect of either proteins or metabolites, would help in this aspect as well. Yeah. So perhaps we could bypass the sorting step at some point and, get the direct information from the serum metabolome data sets, perhaps. Yeah.
Oh, we have a question from a student, Felicia, from Indonesia.
Very beautiful name.
Thank you. So yeah, I'm still an undergraduate student, and I don't really work much about the omics, but in one of my course lecture, in one of my course, my lecturer said that the metabolomic itself is like a complex field of study, because like its dynamic nature. So after listening to your talk, I wonder that how can the advance in like mass spectrometry and other analytical techniques can be applied to overcome the technical challenges of studying the immunometabolomics, like on a cellular or subcellular level.
Yeah. So if I'm not mistaken by your question, you asked that how the mass spec would be employed to study the immunometabolic responses, right?
Yes, because like at first I think you mentioned that the mass spec is also used to study the proteomics.
Yeah. So yeah, maybe I didn't make that clear enough. So mass spec can also be used for metabolomics as well, but it really depends on many factors. For example, the research questions, because some molecules, some metabolites, cannot be well detected using GC or LCMS, but it can be better detected using NMR or vice versa. So that would be one factor. The other factor is about the size of or the volume of the samples. Because some study conducted in animal studies, right? So usually researchers would have very limited volume of samples from the animals, right? So I would not recommend such a study to use NMR, because NMR requires higher volume for analysis, for example. Or some studies collected the samples into the containers where it has the interference compound to the MS signaling. I wouldn't recommend that to be used for LCMS analysis. So it really depends on the convenience and suitability of the samples, and also the project and research questions. So I can't really say which one is the best, but we need to think around the research questions and also the characteristics of our study and also the sample itself. Yeah.
Yeah,
Okay, thank you so much.
To help elucidate that question is like, I think the type of mass spec for metabolite might be different from the one we use for protein as well, right? So there's different type of mass.
Yeah, so especially the mass detectors that we use for proteomics and metabolomics are quite different, and also the setting, and also the elute pump for LC-MS, for example, we use different pressure and different types of elute pump. You know, there would be some minor differences that we need to be concerned. Yeah.
Okay, thank you very much, Ka. So we already like have 15 minutes past. So thank you, Ajahn Jutarop, very much. And you, as you see, his Phenome Center have NMR and mass spec and all the facility to look, you know, for those of you who are interested in metabolite or metabolomic, you can contact him. And I will, you know, share his web, the workshop that he, you know, advertised in our Facebook and in our website. So for those of you, especially students, can apply and to join his more detailed workshop in January. So if there are no other questions, I would like to, on behalf of Thailand Hub of Talent in Cancer Immunotherapy, I would like to thank Dr. Jutarop very much for taking your time to, you know, be with us today, and hopefully we will talk with you more and have a research project with the cancer immunotherapy groups with you. Thank you very much, Ka.
Thank you so much for having me. Have a great day, everyone.